Welcome to another deep dive. We're diving into cancer treatments today, the quest for ones that are more effective, A&D, less damaging to the body. Like a precise scalpel, not a sledgehammer.
Absolutely, a goal we're always striving for.
Right. So this deep dive, it's on a study that really caught my eye. The study focuses on this substance, PM701, and how it affects a specific type of human lung cancer cell. And get this, it suggests PM701 might be able to eliminate those cancer cells while actually helping healthy cells thrive.
Wild, right? It's a very intriguing concept. You see, many current cancer treatments, they do resemble that sledgehammer, impacting healthy cells along with the cancerous ones. That's a major reason for those harsh side effects. So the selective action of PM701, if proven, could be truly revolutionary.
Okay, so before we get ahead of ourselves, we need to clarify something for our listeners. This initial study, it was done in vitro, meaning it was in a controlled lab setting, not inside a living organism.
Exactly. Think of it as laying the foundation before building a house. You need to make sure the base is solid.
So it's a crucial first step.
Yes, precisely. In this case, they used three main types of experiments to really examine PM701's impact: fixed and stained cells, a live experiment, and cell counts.
Multiple perspectives, like looking at something through different lenses.
Exactly.
Yeah.
Each approach offered a unique perspective on the same phenomenon, strengthening their findings.
Makes sense. So let's break down what they saw. Starting with the fixed and stained cells experiment. What exactly does that entail?
Okay, so in this method, they treat the cells with PM701 for different time periods. It's like taking snapshots of their progress over time. This lets them compare the appearance of the cancer cells exposed to PM701 with those that weren't. They're looking for any signs of damage or changes in their growth patterns, really anything out of the ordinary.
So like time-lapse photography, but on a microscopic level, that's fascinating. And then they took it a step further with the live experiment, right? What did they observe there?
This is where things get really captivating. They were able to observe the effects of PM701 on both the cancer cells and normal skin cells, called fibroblasts, in real time. What they witnessed was striking, especially with the cancer cells, and it happened really quickly, even within minutes of introducing the PM701.
Whoa, I bet. Before we get into those changes, though, can you clarify something? Why include skin cells in a study about lung cancer?
Great question. And it highlights what makes this study so potentially groundbreaking. Remember the sledgehammer analogy?
Targeting everything versus being precise.
Yes. By looking at PM701's impact on both the lung cancer cells and the normal fibroblasts, they could see, does it target just the cancer cells? Or does it harm all types of cells?
So it acted as a control, a point of comparison.
Exactly. They were looking for that precision we talked about earlier.
I see. That makes a lot of sense. Okay, so we've got these snapshots, we've got live footage. I'm guessing that third experiment, the cell count, is where they get some hard numbers.
You got it. It's all about quantifying the effect. How many cells remained alive after being exposed to PM701?
Like a microscopic head count. And what did those numbers reveal?
Well, they observed both the cancer cells and those normal skin fibroblasts in these experiments, right? And the differences were remarkable. In the fixed and stained images, the cancer cells exposed to PM701, they showed these, noticeable signs of distress over time, almost as if they were withering away.
So those snapshots were showing a pretty bleak picture for the cancer cells.
They really were. And then the live experiment, that offered even more dramatic visuals. Within moments of encountering PM701, the cancer cells, they started displaying some, unusual behaviors, like their outer membranes, they became distorted, and their nuclei, the control centers of the cells, they appeared pale and faded, as if they were shutting down.
Wow. Okay, those are some pretty vivid descriptions. So it wasn't just cosmetic. What happened to the actual count of cancer cells as time went on?
Well, that's what the cell count experiment showed us, and honestly, the results were pretty startling. Within days of being exposed to PM701, the number of viable cancer cells, it plummeted dramatically, significantly less compared to the groups that weren't treated with the PM701.
Wow, that is a huge difference. So it sounds like the PM701 was effectively wiping out the cancer cells. What about those normal skin fibroblasts, though? Did they suffer the same fate?
That's where the really exciting part comes in. Unlike the cancer cells, the normal skin fibroblasts, they not only withstood the PM701, but in a lot of cases, they seemed to flourish. The live experiment actually showed them continuing to grow and spread normally, even with the PM701 present.
That is incredible. So you're saying that not only did this PM701 seem to obliterate the cancer cells, but it also potentially boosted the growth of the normal cells. That feels like a game changer.
It's definitely a promising finding. But we have to remember, these are still very early results from one single study. We can't draw any definitive conclusions just yet.
Of course, always have to keep our scientific hats on. So where they go from here? What's the next step in this research journey?
That's a great question, and to answer it, we need to remember this study was done in vitro. It's a world away from the complexities of the human body.
Right. It's a controlled environment, not the intricate system of a living organism.
Exactly. So the next step would be conducting more research, but this time using a variety of cancer cell types. We need to see if PM701 has that same selective power over other types of, well, bad guys within the body.
That makes a lot of sense. But let's say they do find consistent results across different cancer cell types. What happens then?
That's when things get even more complex, and the research takes a big leap. If the PM701 keeps showing this level of promise in further in vitro studies, the next logical step would be to move on to animal models, like mice, for example.
Because even though it had a positive effect on the normal skin cells in this initial study, we need to know for sure that it's not going to cause any unforeseen side effects on other cell types, tissues, or organs, right?
Right.
In a living, breathing organism.
Precisely. Moving to animal models allows researchers to observe how the substance behaves in a much more complex biological system. They'll learn things like absorption, distribution in the body, how it's broken down, and any potential toxicity. Basically, does it do any harm?
Right, so it's about moving from a very controlled setting to one that's a lot more representative of how a potential treatment would actually interact with the human body.
Exactly. And while this research is definitely exciting, it's really important to remember that this is just the very beginning of a very long and a very meticulous process.
Of course. There's a reason drug development takes years, sometimes even decades. It requires a ton of research and testing to make sure it's both safe and effective.
Absolutely. But even with that cautious outlook, this research offers a really compelling glimpse into a future where cancer treatments, they might become much more precise, less toxic, and ultimately, even more effective.
It really is remarkable. And this study, it sparks so many questions. For instance, if this PM701 has this incredible ability to target and eliminate cancer cells, how does it even work? What's happening at the molecular level?
That is the million-dollar question. Unfortunately, this particular study doesn't give us a definitive answer to that. While they observed the outcomes, those precise biological mechanisms, there's still a bit of a mystery.
So for now, PM701 remains somewhat of an enigma. We've seen what it can do, but its origin story, that still eludes us.
I like that. A very apt analogy. And like any good mystery, there are bound to be twists, turns, and yeah, maybe even a few setbacks along the way. But that's science, isn't it? It's all a journey of exploration.
Definitely keeps things interesting. Is there anything else from this study that stood out to you, something our listeners should keep in mind?
Yeah, there is one more detail I found interesting, and it really highlights how crucial dosage is with these potential treatments. While this PM701 seemed like a superhero at lower concentrations, the study showed that at very high concentrations, it could actually become toxic, even to the normal, healthy cells.
Oh, wow. So it's all about finding that sweet spot, that balance between effectiveness and safety. Too much of a good thing, as they say.
Exactly. This finding really emphasizes why doing thorough research and figuring out the right dosage is absolutely paramount in drug development.
Makes complete sense. So after diving into all of this, what's the key takeaway here? What does this all mean in the grand scheme of things? So we've seen this research, while early, is super promising. But let's step back for a sec for our listener. How does PM701's approach, how is it different from what we have now? What makes it so groundbreaking?
It's a question at the heart of it all, really. See, many cancer treatments today, they work by going after rapidly dividing cells, which makes sense on the surface.
Cancer cells, they divide quickly.
Right. But the problem is that includes healthy cells too, cells we need for things like hair growth, our immune system, healthy digestion.
It's like trying to get rid of weeds by just dousing your whole garden with the strongest weed killer you can find.
Exactly. You might get rid of the weeds, sure, but at what cost? What we're seeing with PM701, at least initially, is it seems to act differently. It's like it's targeting something specific to those cancer cells, leaving the healthy cells, the ones that those other treatments often harm, untouched.
It's like that image of a gardener carefully plucking out weeds by hand. Instead of this scorched earth approach, it's targeted. We minimize the collateral damage.
Exactly. And that's what's so exciting.
So let's imagine down the line, PM701, it pans out. It becomes a real treatment. How could this change how we think about cancer care?
Well, for starters, just imagine the impact on patients' quality of life during treatment, those harsh side effects.
Yeah.
The hair loss, nausea, weakened immune systems, if we can avoid those.
It's almost hard to imagine for people who've been through it or who've watched loved ones go through it. Those side effects are almost seen as inevitable.
Right. And with this, they might not be. But it goes beyond that. Faster recovery times, fewer long-term complications for survivors.
So shorter treatment, quicker return to normal life, fewer health problems down the line from the treatment itself. That really could change everything.
It's a whole new paradigm,
Mhm.
potentially. But a vital point. We're still early. This is one study, lots of unknowns.
Of course, we can't overstate that, so let's address that. This study, it didn't explain how PM701 works. It's a black box, in a way. Why is figuring that out, the mechanisms at play, why is that so hard?
Well, think of it like this. We're trying to decipher a language, but it's being spoken by these tiny microscopic things. It takes incredible precision, sophisticated tools. It's not just watching what happens, it's figuring out the why, the how on a molecular level.
Understanding the dance of molecules, not just seeing the end result.
Exactly. To really get into the nitty-gritty of PM701's mechanisms, well, we'll be looking at its chemical structure, how it binds to molecules within those cancer cells, the chain reactions that cause those cells to die.
A huge puzzle with tiny pieces.
Mm.
But if we're seeing these results, why does it matter so much to understand how it works?
Because that understanding, that's the key to unlocking its full potential. Can we make it even more effective, target specific cancers better? And are there risks we don't see yet? Maybe how it interacts with certain proteins or pathways in the body could reveal things like potential drug interactions or unforeseen side effects down the line.
Maximizing the good, minimizing any potential harm.
Right. Moving from it works to here's precisely how W and WHY and what else we can do with that knowledge. That's how a promising finding turns into a truly effective and safe treatment.
It's like knowing the how unlocks the full potential of a discovery, especially with medical treatments.
Absolutely. And this ties into something else that's crucial in drug development, and that's dosage. Like we were saying, finding that sweet spot, the balance between effectiveness and safety.
Yeah, the study even showed that. Too much of a good thing, even something that seems super beneficial at one dose.
Can turn harmful, absolutely. That's where things like preclinical and clinical trials are so vital. It's not enough to know PM701 can take out cancer cells. It's what's the right dose. And not just any right dose, but the optimal one for each person, taking into account their age, their health overall, the specific type of cancer they have, even the stage it's at.
So really tailoring the treatment to the individual, making it as effective as possible, but also as safe as possible.
Exactly. A delicate balance, no doubt, but that's what we're aiming for. Personalized medicine, where we can really minimize the risks while maximizing the benefit.
It's amazing to think about how far we've come, from kind of blindly targeting any rapidly dividing cell to this. Being able to go after specific cancer cells, leave the healthy ones alone. It feels like a turning point in how we approach cancer care.
It really does. PM701, it's just one piece, sure, but it's a good example of this shift we're seeing toward precision medicine. It's not just fighting a disease, it's understanding how each person's body works, their own biology, and working with that.
Incredibly hopeful, especially for people facing that diagnosis and their families. Well, expert, this has been fantastic. Thanks so much for taking this deep dive with me.
My pleasure. These kinds of discoveries, thinking about what they could mean for the future of healthcare, that's always exciting.
It really is.
Yeah.
And to our listeners, we hope you've enjoyed this deep dive as much as we have. It's a journey of discovery, this quest for knowledge, and we're glad you're on it with us. We'll see you next time for another fascinating exploration.
Love what you're hearing? Hit that subscribe button, drop a like, and share it with others. Your support fuels the journey for more awesome content.